Alright, everybody, welcome to today's session for your Human Growth and Development online course. We've finished all the basic sessions, and now we're going to actually get into the development and all the changes that happen throughout that process. And what we do is we divide the development process into weeks. We're going to take the first week, second week, and third week, and then we're going to look at the different periods of development. So let's get started. Okay, so the first week of development is actually considered from the point of ovulation. Okay, so anything that takes place after ovulation, and of course, in the case that pregnancy actually does happen, is considered part of the first week. And as you guys know, ovulation is the release of the ovum from the ovary into the uterine tube. And what happens is that once it reaches the uterine tube, then obviously the ovum needs to actually start traveling through the fallopian tube. It can't just stay at the end of it. So there are two things that help the movement of the ovum through the uterine tube. So first of all, it is received by the fimbria, which, remember, were the finger-like projections at the end of the uterine tube, the ones that were found directly over the ovary. And then internally, the wall of the uterine tube has what we call cilia. Okay, cilia are basically hair-like projections. Think of them like a hairbrush, okay, or the broom they used to sweep the floor. Girls, you know what I'm talking about. Okay, so what they're going to do is they're just going to sweep or move the ovum further into the uterine tube. And the other factor that helps with movements of the ovum is contraction of the muscular walls of the uterine tube. They also help carry the ovum down towards the uterus. So those are the two things, the sweeping action of the cilia and the contractions of the muscular wall. They're the ones that are going to move the ovum further down into the uterine tube. Okay, so this is just a quick picture to show you the process of ovulation. These projections here are the fimbria. Okay, and then you can see small elevations or projections through the wall of the uterine tube. These are not the cilia, okay, but this is normally where they're found. And then along with the muscular contractions, the ovum is going to end up from here, and then it's just going to start moving down the uterine tube on its way to the uterus. Now, the second event of the first week of development is fertilization, which at this point we all know is the fusion of the male and the female gametes. And this has come up before, but let's just say it again. It occurs in the ampulla of the uterine tube, which we've already mentioned is the widest part of that organ. Now, fertilization has certain results. So basically, once this process happens, there are certain changes that take place. The first change is the formation of the zygote. Okay, so remember we have the separate ovum from the female, we have a separate sperm from the male, but once they fuse together during fertilization, the resulting cell from that process is now called a zygote, so it has a completely different name. The other thing that happens is restoration of the diploid number of chromosomes. Okay, so remember we have two haploid cells, they fuse together, half the number of chromosomes from here, half the number of chromosomes from there, put them together, and you end up with a diploid number of chromosomes. The third thing that happens is determination of gender at the genetic level. Now, remember that chromosomes exist in pairs, and you have a pair that is found at the end of the genetic map or in the genetic diagram called the sex chromosomes, okay, which can either be an XX chromosome if it's a female or an XY chromosome if it's a male. So obviously once fertilization happens and the chromosomes start arranging themselves and then you end up with the diploid number of chromosomes, then the sex chromosomes are going to be in position. So that's what we mean when we say that the gender is determined at the genetic level. However, this is different than when a lady is pregnant and then she tells the doctor that she wants to know the gender of her baby through the ultrasound, because that requires a later stage of development when the reproductive organs have actually started developing and the doctor can see the reproductive organs through the ultrasound. So those are two completely different things. Okay. Now, what we're looking at here is a picture of the sperm head. This is the head of the sperm here, or actually this whole thing here. All right, and this is the nucleus, this light blue structure here, okay? And yeah, that's where the chromosomes and genetic material is going to be found. So I want you to notice that at the tip of the head of the sperm, there's like this yellow covering, okay, that kind of protects the head of the sperm and then protects the nucleus as well. Okay. Now this layer is made of glycoprotein. Okay, and it is called the acrosome. All right. Now, since it is a glycoprotein layer, and then obviously you can see here that it has an outer membrane and it has an inner membrane. Okay, so this structure kind of acts like a barrier or protecting layer for the sperm, but we need this to be removed, okay, in order for fertilization to happen. Because if you notice, it's kind of like a barrier between. So if the ovum is here, where this box is, then obviously the nucleus of the sperm and the nucleus of the ovum will not be able to meet because there's this barrier in the middle here. Okay, so we need this layer to be removed, all right, in order for the sperm and the ovum to be able to fuse together. So if we move over down here to the third picture, okay, you can see how the glycoprotein layer has been removed. The only part that's left over is the inner membrane, and there's just a little bit at the side here. Okay, but generally now the nucleus is uncovered, and fusion with the ovum can occur. Now, this process, okay, is called capacitation. Okay, so before the sperm is able to fertilize the egg, it needs to undergo a series of changes that involve removal of the glycoprotein layer lining the membrane at the tip of the sperm head. Okay, this is called the acrosomal region. This process happens in the uterine tube, so remember, it doesn't happen once the sperm is released from the male reproductive organ. It doesn't happen in the cervix, it doesn't happen in the uterus. It has to wait until the sperm reaches the uterine tube, and that's when it starts. And it generally takes 7 hours, and like I said, it's called capacitation. So removal of that yellow layer is called capacitation. Now, once that layer has been removed, okay, another reaction takes place called the acrosomal reaction. Now, this involves the release of penetrative enzymes, basically enzymes that can break down the layers or the outer layers of the ovum in order to allow the sperm to get further down or further into communication or contact with the ovum. Okay, so those are the two things that happen: the capacitation, which is the removal of the glycoprotein layer on top of the sperm head, and the acrosomal reaction or the acrosome reaction, which is release of penetrative enzymes in the acrosomal region. Okay, so that's in terms of the sperm part of fertilization. Okay, now what we're looking at here is a picture of the ovum, and I kind of thought this was cute. The nucleus looks shocked for some reason. So before we move further into the lecture, okay, we just need to go over the structure of the ovum real quick. Okay, so this pink part is the cytoplasm, where you can find the different organelles, and you've got the nucleus. Okay, now notice that it has a little bit of a thick outer lining, okay, just outside the cytoplasm. This layer is called the zona pellucida. And then there's another layer of cells, which are the leftover cells from the from the granulosa cell layer. Okay, and this outer layer here, once it's released at ovulation, becomes called the corona radiata. Oh wow, corona, I just realized. Okay, so you've got the zona pellucida right outside the cytoplasm, and you've got the corona radiata outside the zona pellucida. Okay, so remember the sperm, in order to reach the nucleus of the ovum, has to penetrate all of these layers. And that's where the penetrative enzymes from the acrosomal region come into play. So the process of fertilization actually happens in phases, okay, and obviously each phase is going to have a specific event. So the first phase is penetration of the corona radiata, which was the first layer that the sperm would encounter if it's trying to fertilize the ovum. And then logically, what would be the next phase? Okay, penetration of the zona pellucida, naturally. Okay, and then the outer membranes of the oocyte and sperm cell will fuse together. And then the last phase is fusion of the nuclei of the egg and the sperm. All right, so essentially it happens in 4 phases. Penetration of all the outer layers of the ovum, and then fusion of both their membranes, and then eventually fusion of the nuclei. Okay, so what we're going to look at here is just everything I just said in animation form. And before I move forward, let me just emphasize this video is not mine. I got it from a YouTube video and downloaded it. So, and I just use it for teaching purposes. Okay, so that was the process of fertilization. Just to be clear, I know there were a lot of labels and things that were mentioned on that video. Again, I don't need you to go into too much detail or remember all of those names. The whole purpose of the video was just to allow you to see it in animation form. Okay, let's keep going. All right. So we're just going to look at something called the cortical reaction, which if you guys go back to the video was actually touched upon real quickly. All right. So essentially what we need is for one sperm to fertilize the ovum. Okay, but you could use, like you saw in the video, there were a lot of sperm that reached the egg. So what needs to happen is that once the egg is fertilized by one sperm, there needs to be a specific reaction that prevents the other sperm from penetrating the ovum as well. Okay, and this reaction is called the cortical reaction. Okay, so if you look at, where's my cursor? Okay, if you look at the picture on the left here, okay, you can see that within the outer layer or close to the membrane of the ovum, okay, there is something called the sperm binding protein, which is basically the protein that helps attract the sperm to the egg in the first place, okay, because it's going to end up binding to it. All right. And what you have are these granules, okay, called cortical granules. They're just kind of found in the cytoplasm, and then once the process of fertilization or the contact with one sperm takes place, these granules are activated. And what they do is they travel near the surface or the outer layer of the cell, and what they do is, if you notice on the inside of the granule, there's like a yellow circular content, okay? What happens is these cortical granules burst open and they release their lipid contents, all right? And what those do is they end up forming a jelly coat or impenetrable layer outside the ovum, all right? So that if we move on to the next picture, you'll notice that number one, the structure of the outer lining of the ovum has gone from curvy to straight. Okay, so that's the jelly coat that was formed by the cortical granules. And the other thing is that the sperm binding proteins have actually disappeared. So any other sperm that comes and tries to fertilize the ovum will not be able to, because the things that allow it to penetrate the outer layers of the ovum are now gone. So this process is the cortical reaction. So the cortical reaction prevents what we call polyspermy, and polyspermy by definition is fertilization of one egg by more than one sperm. The cortical reaction is part of the processes that prevent polyspermy, which means that it's not the only reaction, it's just one of them, that prevent polyspermy from happening, and it starts as soon as fertilization begins. It is the function of specialized granules from the cytoplasm of the ovum called the cortical granules, like we saw in the picture. Cortical granules fuse with the membrane of the ovum, and then they release their contents outside the cell, building up a layer that can no longer be penetrated by the sperm. Okay, so remember fertilization, and as soon as it starts, you have initiation of the cortical reaction. Okay, so now we are at the stage of the zygote. All right, so you've got the sperm, the egg, they fuse together, they formed a single cell that is now diploid, and it is called the zygote. Okay, now the whole idea is that I need this single cell to somehow become an embryo that can then develop into a fetus, that can then develop into a human being. Okay, or a fetus that becomes a human being. So what's going to happen is that this single cell is going to undergo a series of mitotic divisions, okay, in order to increase the number of cells that I have. So this single cell, once it's been fertilized, it's going to undergo something called cleavage. And the first cleavage gives me 2 daughter cells. And then each of these is going to undergo another division. Okay, so automatically I'm going to end up with 4 cells. And then the 4 cells will give me 8, and then the 8 will give me 16. Okay, so each time I have a cellular division, I basically end up doubling the number of cells that I have. Once the zygote has divided enough times for it to have 16 cells, I change its name and I now refer to it as a morula. Okay, and then we're going to see later on how the morula undergoes another set of changes in order to give me something called a blastocyst, and then that's going to undergo changes to give me the actual embryo. So it's a cycle, all right, and each phase has its own specific process. So the zygote undergoes a series of mitotic cell divisions, like I said. The first single cell directly after fertilization will divide to give me 2, and then the 2 become 4, 8, and 16. And when it is at the 16 cell stage, this is usually 3 days after fertilization, this is referred to as the morula. Okay, so we're just going to look at that in animated form again. Okay, we're going to stop there because anything that comes after this point is our next session. We don't need to view this right now. Okay, so I just hope that you guys saw how it divided into a different number of cells each time. And the thing that I love about that video is the very dramatic music. It makes me feel like I'm in an action movie or something. All right, so that was cleavage. All right. So now we're at the stage where the cellular division and cleavage has happened. The morula has undergone certain changes, and now we have the process of implantation. Now, implantation is when the developing embryo is embedded in the uterine wall. Now, this occurs 6 days after fertilization, guys. Please make sure you remember these days and times, okay, because they do tend to come up in the exam. Okay, so implantation, which is attachment of the embryo to the uterine wall and its embedding into the endometrium, happens 6 days after fertilization. Normally, implantation should happen in the body of the uterine wall. It can either happen in the posterior, which is the back wall, or in the anterior wall or anterior part of the body, which is at the front. Okay? Commonly, it's in the anterior wall, but it can happen in the posterior wall as well. Okay? So, with this diagram again, you've just got all the different stages we've been talking about since the beginning of the lecture. Okay, up until the point of implantation. Okay, so this is when the ovulation happened and the fertilization took place here, and then the cellular division started at this point, the cleavage. Okay, it's a morula here, so that means it has 16 cells. Okay, notice how there's a few cavities and things showing up. We're going to talk about those later. Okay, and then here you can see how the developing embryo is actually in the endometrial lining. So this is the endometrium, this is the myometrium, and this is the perimetrium. So the embryo is actually embedded inside the endometrial layer, and this is the process of implantation. Okay, so let's just look at implantation real quick. Okay, so with that video, you guys, again, I know there were a lot of new things that kind of popped up. All right, don't worry about them at this stage. Okay, all I wanted you guys to see with that video was the passage of the developing embryo into the endometrial lining, which is the process of implantation. The rest of the things and all the other changes, we're going to get them next session, so don't panic. So, in summary, the major events of the first week were ovulation, which was release of the ovum, the mature ovum from the ovary. Fertilization, which was fusion of the ovum and the sperm that happens in the ampulla of the uterine tube, and all of its subsequent events. So the cortical reaction and everything that happens after that. Remember, once the zygote is formed, it undergoes a series of cellular divisions in the form of mitosis to go from a single cell stage to a 16-cell stage, which was referred to as the morula. And this series of cellular divisions is called cleavage. And then finally, at the end of the first week, you have implantation, which was the embedding of the developing embryo within the endometrial lining of the uterus. And that concludes our session for today. Those are the major events and things that you need to know about the first week of development. And as always, I'm going to leave you with a positive quote. Remember, I know these are difficult times, you guys, so remember to take it easy on yourselves, and it's okay if you fall down and just, have one of those bad days, but just make sure that when you get up, you rise as the whole damn fire. Thank you for joining me. I hope this session was useful, and I will see you guys next time.